Good afternoon. First, I would like to thank the DIM to give me the opportunity to present our work. So I won't speak about bacteria or vector. I will speak about EV, extracellular vesicle, because I think there are some people here who do a lot of EVs. So I will present you some data about the potential use of EV loaded with different RNA, that could be miRNA, siRNA, or RNA, to treat aggressive cancer. And aggressive cancer for us means cancer that can develop a lot of metastases. Okay, so indeed, as you know, in cancer, there is a gene deregulation, and most of the time, many genes are deregulated. It could be mutation, it could be signaling modification, many modifications. But the problem at this time, it's in fact the therapy are very limited because sometimes they target only one gene, and so that you have an incomplete response. So where we should go, in fact, we should go to, I try to find, there is a pointer. Il marche pas. Ah, okay. Okay. So, in fact, where we should go, it's to find a therapy that will be able to target many genes at the time or many pathways to be able to have a complete response. And the way we can do that, it's in fact using EVs, extracellular vesicle, that we will load with different miRNA that will target different genes, different pathways, or EV loaded with different siRNA directed against different genes. And in fact, why we propose that, and what is the origin of our work and of the developing of this potential therapy? In fact, we have recently shown that cells that express transcription factor NFAT3, it's a transcription factor we have shown is anti-invasive, anti-tumoral. These cells are able to produce EVs that are anti-tumoral. Indeed, these EVs are able in vitro to block cell proliferation. So here there are some results about blocking the proliferation of breast cancer cells, able also to block cell invasion in vitro. And in vitro, these EV that are endogenously antitumoral are able also to block melanoma invasion, glioblastoma, and pancreatic cancer. We then went further to try to see if in vivo this EV could have an effect, and indeed we have shown in a breast cancer model that in fact these antitumoral EVs are able to block metastasis erasing and tumor growth. We have shown that in fact this EV do not transfer the NFAT3 transcription factor that is antitumoral and anti-invasive. In fact, we have identified the mechanism that is NFAT3, in fact, direct the presence of different miRNA in these EVs. And these miRNA are either anti-invasive, anti-proliferative, or both anti-invasive and anti-proliferative. So we have validated this function of these different miRNA. And in fact, there are, in these EVs, 15 miRNA. So we call this combination miRNA COM15. And the idea was to produce EVs, therapeutic EVs, loaded either with this combination of these 15 miRNA that target many pathways, or either loaded EVs with target gene that this mRNA target. So we try here today, I will just show you results about the mRNA loading of EVs. But we are still working on the siRNA loading with target gene of this combination. But as you know, we have 15 miRNA, and we have to put that in an EV at the same time. So we cannot produce a cell that will be able to produce all of these 15 miRNA. So the only way we can load these EVs with this combination of 15 miRNA was the exogenous loading of this mRNA. So we produce the EVs, and then we treat them to be able to load them with this 15 mRNA. And here is shown the protocol we use. It's a protocol that has been published by a team directed by Stephen J. And in fact, it's a protocol based on the deshydratation of the EVs. We produce EV, we deshydrate these EVs, and then we put it in a citric buffer, an acidic buffer, and then after we dialyze to be able to have some proton on these EVs inside and outside. And then we incubate these EVs with mRNA, and the mRNA either enter the lumen of the EV or stay bound to the membrane. And after that, we wash away the mRNA that didn't enter the EVs and purify the EVs to use them as a potential therapeutic EV. This method of EV loading work efficiently. Indeed, we have an efficient loading of EV in mRNA, and we have defined the higher dose we can use. That is here, for example, 400 nanomolar. And if we put more mRNA, we won't go further in the loading. If we look at the effect of this EV loaded with this combination of mRNA, we have a dose-dependent effect on the growth of here breast cancer cell. So we have a dose effect. And we have decided, in fact, to compare this EV mRNA loaded to other EV. We use EV from human ADSC. It's human adipose stem cells that are widely used by many people in many conditions. And we load also this kind of EVs. And as a control or gold standard, we use also liposome that we also loaded with this combination. And in the three cases, we succeed to efficiently load the EVs or liposome with this combination. And in vitro, we have shown that in fact, EVs from HEK loaded with this combination or the EV from the human adipose stem cell are able to inhibit cell proliferation. Then we decide to test it in vivo. So these in vivo data are preliminary data, but they are very encouraging because they show that in fact EVs from HEK, the 293 T cell loaded with a combination, is able to inhibit significantly tumor growth. But surprisingly, the EV from ADSC that are loaded with the same combination of mRNA was not able to inhibit tumor growth. And the same thing for the liposome. And this inhibition of tumor growth in mice work either when we do an intravenous injection or an intratumoral injection, meaning that in fact, even by intravenous injection, this EV, as for now we don't know, succeed to reach the tumor and to have an effect to the tumor. So here it's shown the significativity of this effect of the EV loaded with the mRNA COMB15, sorry. But we have a big problem. So it's why if in here some people can help us, it will be really helpful. In fact, we succeed to load this mRNA, as I said, with an efficient loading, but we lose a lot of extracellular vesicule when we purify at the end after washing away the mRNA that didn't enter the EV. We lose a lot of particles, a lot of EVs. And as for now, we didn't find a way to avoid this loss of EV. And so it will be interesting to collaborate with people who are more used to do bioproduction of EV or purification to maybe find a way to optimize this part of the project. And as you can see, when we begin with 10.9 particles, we end up at the end with 10.6 particles. It's still enough for our assay, but it's not really optimized. So if we want to go, we go further and to go eventually to a therapy, we should improve this step. So really, if you know somebody who, if there is somebody who can help us, they are welcome. I think you understand that we are looking for that. And so it's why don't hesitate to contact me to discuss and maybe to improve this thing, because the therapy seems to work. We still have to improve the biology party, but this party of production is really important. And in fact, it combines what we talk about today. It's fundamental research that might go to the development of a therapy and a startup. So I need some help. And we have a lot of help with the people who has worked on this project. So there are actual members, and there is Genole who presents a poster. It's 39 outside if you want to speak with him. It's a PhD student who work a lot, who load these EVs. And I thank all of the members, the past members too, and the League and ARC, Arganeo, and all the foundations that give us some money to push forward this project. Thank you for your attention.
Yes, one question.
Thank you for the talk. So you showed that cancer cell-derived EVs are more effective for cancer treatment than adipose cell-derived. Do you think that after your method of cleaning, dehydration, and so on, you still have some cancer cell, tumor cell specific molecules on these vesicles that might bring them more effective to the tumor niche rather than the other ones? Because the tumor cells, they use these to form the niche formation for the metastasis. So do you think you still might have some molecules there?
So I will answer yes, but our first study we show that effectively EVs produced by cancer cells that express NFAT3 are able to be antitumoral, but maybe I didn't tell it strong enough, but now we use EV from HCT293T cell that endogenously express NFAT3 and have the same effect. So we don't use any more cancer cell to produce these EVs. And in fact, we choose to switch, and we were lucky that it worked with this cell too, because they express NFAT3 endogenously, with the idea or maybe developing a therapy and to go about to think about the regulation too, that injecting EV from a cancer cell would be maybe not so easy.
Any other question? Or maybe I have one short question, short answer. Do you have an idea of the target of the microRNA?
Yes. Yes. We have identified the gene in the 3 cell lines, the 231, the 2 triple cell line, and the pancreatic cell line. And we are actually working on this gene, and we have discovered new genes that were not known in cancer. So it will be the next step.
Thank you very much. You can discuss during the break, but we are going to stop for the question now. Thank you very much.